Gene mapping describes the methods used to identify the locus of a gene and the distances between genes. A central task of gene mapping is to place a collection of molecular markers onto their respective positions on the genome. Today, we will cover 5 topics. First, we will look at chromosomal recombination. Here is a pair of homologous chromosomes. Locus is a specific position on the chromosome where a gene is located. Allele is an alternative form of a given gene. For example, at this locus, there are 2 alleles. Same for locus 2 and locus 3. Haplotype is a combination of alleles on a single chromosome. For example, D, capital M, C1 is one haplotype, and capital D and C2 is another haplotype. Genes on the same chromosome are syntenic. Assume 2 loci on 2 different chromosomes. Each has 2 alleles. During meiosis, 2 different chromosomes will line up on the metaphase plate in one of the 2 orientations with equal likelihood. After meiosis, there will be a 4 possible combinations of alleles in a gamete. Each combination is as likely to occur as any other. This phenomenon is known as independent assortment. Here are 2 homologous chromosomes. When there's no crossing over between these 2 loci, there's no recombinant chromatids. But here on the right, there's a crossing over between these 2 loci. This results in 2 recombinant chromatids and 2 non-recombinant chromatids. Here is a pedigree. This man in generation 3 inherits a maternal chromosome that contains segments derived from all 4 chromosomes of his maternal grandparents. Therefore, each chromosome a child inherits from a parent is never exactly the same as either of the 2 copies of that chromosome in the parent. Rather, each chromosome contains some segments derived from that parent's father and other segments from that parent's mother. Now, let's move to part 2. We will look at recombination frequency. Map distance refers to distance between 2 loci on the same chromosome. Map distance can be very large when the 2 loci are very far apart. It can be small when the 2 loci are very close, or somewhere in between. The unit for map distance is centimorgan. Map distance can be determined by measuring recombination frequency. Map distance sometimes is called genetic map distance or genetic distance. Here, solid line represents relationship between map distance and recombination fraction. Dotted line represent hypothetical linear relationship between map distance and recombination fraction. On the x-axis is map distance in centimorgan. On the y-axis is recombination fraction. When map distance is below 10 centimorgan, these two lines almost merged. Map distance is in proportion to recombination fraction, with each 0.01 of recombination fraction corresponding to 1 centimorgan. Above 10 centimorgan, these two lines diverge. No matter how far apart two genes may be, the maximum recombination frequency between any two genes is 50%. When genetic distance between two loci is over 10 centimorgan, recombination fraction is not an accurate measure of genetic distance. For example, between loci A and H, recombination fraction is 0.15, and we know genetic distance between A and H is over 10 centimorgan. So the recombination fraction 0.15 cannot be an accurate measure of the genetic distance between A and H. To measure genetic distance between A and H accurately, we should use more markers to further divide this region into smaller regions. Then we measure genetic distance of each of these regions and add them up. Here are the results. After we add them up, the map distance between A and H is 20 centimorgan instead of 15 if we purely base on recombination fraction between A and H. Genes with recombination frequencies less than 50% are considered to be linked. For example, these two loci, when they are close enough, crossover between these two loci does not happen frequently. So these two genes are linked. Two genes with a recombination frequency of 50% are unlinked. There are two types of maps used in the fields of genome mapping. One is genetic maps, and the other one is physical maps. Both maps are a collection of genetic markers and gene loci. Genetic maps distance are based on the genetic linkage information. Physical maps use actual physical distance measured in number of base pairs. Map distance based on recombination frequencies is not a direct measurement of physical distance along a chromosome. Physical distance can be measured by nucleotide or base pair. Overall, the human genome is estimated to contain about 3,200 megabase pair, has a genetic length of 3,615 centimorgan. As a result, roughly 1 centimorgan is equivalent to 1 megabase pair. Some factors can alter the genetic versus physical distance ratio. For example, in recombination hotspots, high frequencies of recombination will overestimate physical length. On the other hand, in heterochromatin regions, for example, very little recombination takes place in heterochromatin region. These will underestimate actual physical length. As a result, these three regions are similar in size in physical map, but they are very different in size in genetic map. Now, Let's move to part 3. We will look at markers for gene mapping. Successful gene mapping requires the availability of a large number of highly polymorphic markers dispersed throughout the genome. Polymorphism refers to presence of 2 or more alleles for a gene or a genomic sequence in a population. We have covered this before in the mutation lecture. There are several classes of polymorphic markers. They include restriction fragment length polymorphism, variable number of tandem repeats, short tandem repeats polymorphism, and single nucleotide polymorphism. To detect restriction fragment length polymorphisms, we use restriction enzymes. There are more than 200 restriction enzymes available, and each recognize a specific DNA sequence. For example, EcoR1 cuts double-stranded DNA at the palindromic size GAA-TTC. As shown in this picture, this enzyme cut these 3 locations in the genomic sequence and generate several fragments. These fragments in different sizes can be detected by Southern blot. Differences in DNA sequence generate different cleavage sites for restriction enzyme. For example, here are 2 alleles. First allele contains EcoR1 site, and the second allele does not. When we cut with the EcoR1, the first allele will create 2 fragments, and the second allele only results in 1 large fragment. Variable number of tandem repeats are the results of varying number of mini-satellite repeats in the specific region of a chromosome. Each mini-satellite repeat typically ranges in size from 20 to 70 base pairs. For example, here are 2 alleles for a specific marker. First allele has 6 tandem repeats. Second allele has 4 tandem repeats. These repeats are flanked by a restriction site. These 2 alleles can be distinguished by different size of restriction fragment. Short tandem repeat polymorphisms, also called microsatellites. They are repetitive sequences in which the repeated unit is generally 2 to 6 base pair long. For example, here are dinucleotide repeats. First allele has 5 repeats. Second allele has 3 repeats. These repeats can be amplified by PCR. These 2 alleles can be distinguished by different size in PCR products. Single nucleotide polymorphisms represent a difference in single nucleotide. For example, here are 2 alleles. First allele has G in a specific position. Second allele has a C in that same position. These 2 alleles can be distinguished by DNA sequencing. Detecting the recombination events between loci requires a parent be heterozygous at both loci. And second, we know what alleles are on the same chromosome at both loci. This information is called phase or linkage phase. For example, this person is heterozygous at both loci. This person has 2 possible phases. One is capital D and capital M alleles are on the same chromosome. And second possibility is small d and capital M are on the same chromosome. This information needs to be provided before gene mapping. When two alleles are on the same chromosome, we say they are in coupling. When the two alleles are on a different chromosome, they are in repulsion. For example, capital D and capital M are on the same chromosome. They are in coupling. Small d and small m are in coupling. But capital D and small m are in repulsion. The first step for phase determination is to determine the genotype of a marker. Specific alleles of a marker are derived from microsatellites, SNPs, or restriction fragment length polymorphisms. Here is a pedigree with a specific marker. The marker has 6 alleles, as demonstrated in this gel. For example, this woman has 4th and 5th bands. Therefore, she has allele 4 and allele 5. It is important that students are very familiar with determination of genotype of a marker in a pedigree, as exemplified here for USMLE. Now, let's move to part 4. We will look at gene mapping. Here is a family in which a mutation causing NF1 is transmitted in 3 generations. The genotype of a marker locus is shown for each member. Our task here is to estimate recombination frequency. The woman in the 2nd generation inherits the disease allele in allele 1 from her mother. Therefore, in this woman, the disease allele in allele 1 are in coupling. Disease allele in allele 2 are in repulsion. To estimate the recombination frequency, we need to count the number of children in whom disease allele in allele 1 are not in coupling, or disease allele in allele 2 are not in repulsion. Of her 6 children, only in 1 child, the disease allele in allele 1 are not in coupling. Therefore, the recombination frequency in this family is 1/6. This is an estimated recombination frequency based on data from one family. To obtain recombination frequency accurately, we need to combine data from many families. This can be done using LOD scores. LOD scores are derived from odds. Odds is a ratio of 2 probabilities. It is defined as probability of observing pedigree data if recombination fraction is 0.17 divided by probability of observing pedigree data if recombination frequency is 0.5. LOD is the logarithm of the odds. A great advantage of using LOD is that LOD scores from individual families can be added up together. Regarding NF1, LOD scores from several families have been determined and added together using a computer, and the results is shown here. For example, when recombination frequency is 0.01, LOD score is 0.58. But when recombination frequency is 0.1 and the LOD score is 3.47, the LOD score has a single peak right here. Therefore, the most likely recombination frequency between the gene and the marker is about 10%, corresponding to 10 centimorgan of map distance. When interpreting LOD scores, the following rules apply. A LOD score greater than 3.0 shows statistical evidence of linkage. An LOD score less than -2 shows statistical evidence of non-linkage. A LOD score between -2 and 3 linkage state is indeterminate. Second example is a family with retinitis pigmentosa. It is an autosomal dominant disorder. Here is a pedigree. RP9 is a disease-related gene, and locus 2 is a marker. In this woman, allele B and allele D are in coupling. Small b and small d are also in coupling. These linkage phases remain the same in all of her 6 children. Therefore, recombination frequency is 0, as shown here. Regarding RP9, LOD scores from 3 families have been determined and added together using a computer. Here is the result. The LOD score has a single peak when theta is 0. Therefore, the most likely recombination frequency between RP9 and locus 2 is less than 0.01, with a map distance below 1 centimorgan. Finally, let's move to part 5. We will look at gene conversion. Most of the time, segregation of markers follow the law of segregation. Alleles segregate during meiosis, such that each gamete contains only one of the alleles. Gene conversion is an exception. Gene conversion is a process by which one DNA sequence replaces a homologous sequence, such that the sequences become identical after the conversion event. Conversion of one allele to the other is often due to mismatch repair during homologous recombination. During meiosis, 2 homologous chromatids can pair up and DNA strands can be exchanged between them. For example, these are segments from 2 homologous chromatids. Exchange of DNA strands will lead to formation of heteroduplex with 1 base pair mismatch, as shown here. Mismatch repair is going to fix the mismatch. If the top strand is the native strand and the bottom strand is a newly exchanged strand, there are 2 possibilities after mismatch repair. If T changes to C, the chromatids remain the same identity as the big A allele. But if G changes to A, the chromatids change the identity and become the smaller A allele. So this situation is described here. T to C, no conversion. G to A, changed identity. Big A, changed to small a allele. Using the same method, we can easily figure out the 6 other scenarios. Taken together, identity of a marker can be altered through gene conversion during meiosis